Thank you. How to remove this? Okay, thank you. Okay, good morning, all of you. Once more, pull check whether the sound is going. So today we are going to discuss about defense systems in bacteria. So by this time, you have all come to know more or less about some of the defense systems in some of the lectures delivered by Professor Kanan and Samrat Bora in the morning. He was speaking about some restriction enzymes. Anyway, we'll study in detail about the defense systems in bacteria today. So I will start with very basic concepts for the beginners who have participated over here. Once again, for some of you, it may be some small revision, and I think you will enjoy it. Anyway, so some of the most seminal work of this era around 1840s was the germ theory put forward by Louis Pasteur. All of you know about it. And those who don't know about it, it was one of the most important theory which actually, inspired a large generation of biologists, microbiologists to pursue research in this interesting area, where he put forth the theory of, certain agents called germs, which causes diseases. So let me not go into details of those. He, along with Chamberlain, actually developed one filter, and this filter is known as the Pasteur-Chamberlain filter. So what is the utility of these? We will tell you later. Now, another seminal idea of the time was the cause postulate. Okay, so those who are already experts in pathology know about the cause postulate. There are four important things in each. So whenever you see some kind of disease, the disease, there is some kind of a positive agent. It may be a biotic agent or a biotic agent. But in this case, we are much more focused about the presence of pathogens associated with the disease. So for that particular disease, the first condition is that a pathogen must be found in the disease. Okay, number one condition. The second condition is that this pathogen could be cultured, or it has to be maintained in culture. And these cultivated pathogen, if you take out from the culture and then you inoculate into organisms, whether it is animals or plants in which the disease was caused originally, it will cause disease in that particular organism. And from these disease organism, you can again re-isolate the pathogen and grow in culture. So these all four conditions has to be fulfilled. So this looks like very simple and in a way very boring, but they are very interesting to science. As we'll discuss in the next slide, you will find out why. So let us remember once more what is the cause postulate. Number one condition is pathogen should be found in disease, should be maintained in culture. If you inoculate the culture material into animals or organisms, it will cause the disease. And from this disease organism, you re-isolate the pathogen and grow it in culture. So now for doing this, they use this Chamberlain filter. So when you take the isolates of these pathogen and you filter it, so the pathogen will be retained in the membrane through which you are filtering it. So this membrane retains the infectious material, and that infectious material, in the first instance, when you are isolating it from a disease organism, you take it for culture. And in the second instance also, when you were reinfecting it, you re-isolate and then you again collect it from this membrane. So this is a thing which really revolutionized, very simple idea. It revolutionized biology. So a couple of decades after these, in 1886, Mayer was studying one disease in tobacco because the leaves have a mosaic appearance. So this disease was named as mosaic disease of tobacco. And Mayer did extensive study of this particular disease, and it describes these disease symptoms in detail. And using this cause postulate, he demonstrated that the disease can be transmitted by using the sap from the affected tobacco plants as the inoculum to infect healthy plants. And he thought that this disease was spread by very small bacteria or toxins. That was his assumptions at that particular point of time. But he could not find any signs of fungi or bacteria in the infected sap. Using optical microscopy, this was because it was not known to him what was the causal agent. And it was also not known to him that the causal agent is actually too small to be detected in an optical microscope. So it was not actually something what he thought about, small bacteria. Okay, so he could not detect it. But he made a conclusion, maybe in a hurry or due to some kind of error, that the infectious agent was some sort of bacteria. And then he falsely claimed, basically, or erroneously claimed, that he was able to obtain the clear filtrate in this Pesce-Chamberlain filter from the infected sap using filter paper in several repetitions. So this is one of the most, kind of experiment which is now being cited by people who teach research methodology regarding errors or some kind of scientific interference which is wrongly derived from the experimental evidence. And this was exactly what happened at the point of time. So anyway, Mayer was the first person who did some detailed study on the tobacco mosaic disease, and he described the symptom quite well. He could make some sense of the causative agents, but actually there was no any much scientific substance on the pathological part of the study conducted. In 1980s, few years, 1890s, another person in 1892 exactly, Russian Dmitry Ivanovsky, he reported that extracts from leaf remains infectious after filtration through a Chamberlain filter canal. This was altogether different than what Mayer was claiming. Mayer was telling that the filtrate was causes the disease, but Dmitry showed that the liquid sap that which passed through the filter into the chamber below contained the infectious agent. And then if 6 years or 5 years after that, Beijering, who was not aware of Ivanovsky's work, reported the same thing in the tobacco mosaic disease described by Mayer. And he reported one thing additional to it, that the filtrate was sterile. I mean, it did not contain any kind of bacteria, but it was still infectious. And he published his work in a Dutch and German journal. And he actually used a filtrate, okay, to reinfect not the residue, to reinfect plants, and he could see the symptoms over here. So he proved that it multiplied only in tissue in which cell division occurred. So for this particular infectious agent to get multiplied, you have to infect it into a live and active area of the plant. And he coined the famous term contagium vivum fluidum, which many of you may have heard. And it is now well appreciated that Beijerinck was the first person to use the term virus, which is specified to be liquid or soluble agent that reproduces itself in a living plant. So this is in brief the history of the discovery of virus and how it was found to be infectious. But what was claimed by Mayer was actually found to be false by Ivanovsky and Beijerinck. And as you can see here, the cause principle, which I laid a lot of emphasis in the beginning, they found out that the pathogen or the infectious sap causes disease. Yes, so they're putting 2 positive over here, but they could not culture it. Okay, so they put it no. But if you re-inoculate the material, it causes disease. Okay, that is true. But they could re-isolate it into culture. The answer was no. And then later on, additional things they added up. It was invisible. They could not see it. Okay, until the discovery of development of the electron microscopy. And by the 50s, 60s, the discovery was confirmed that this tobacco mosaic virus basically contain nucleic acids. Anyway, so nowadays, another important development, which is of big relevance to today's lecture, the work by English bacteriologist F.W. Ford. So after this discovery of these bacteria in of this virus in 1915, Ford described a transmissible lysis in a micrococcus. He actually also had to go to the war at that point of time, and there were many other reasons. He had to abandon his discovery. And when he came back to research, he tried to propagate vertebrate viruses and not viruses which, infect micrococcus. So, but anyway, he was the first to describe a transmissible lysis in a micrococcus. 2 years later, Felix de Heril described the lysis of Shigella cultures. And after his long years of dedicated scientific work with bacteriophages, he also thought about the phase therapy of infectious diseases. So since this phage, which is a term he coined as bacteriophage, kills the bacteria, he thought, and he actually worked towards the use of these phage particles in the application of infectious diseases caused by bacteria. So today we know that these bacteriophages are viruses, and they are obligate intracellular parasites that infect bacterial cells. Now, in 1935, story of these tobacco mosaic virus continued, and it was the first virus to be crystallized by Stanley. And in 1946, the Nobel Prize was actually awarded to 3 persons. Sumner got half of the award, whereas Northrop and Stanley got the second half, 1 for each. And this entire work was for Sumner got it for the crystallization of enzymes, while Stanley and Northrop got it for the crystallization of proteins, and particularly Stanley's work on the crystallization of this tobacco mosaic virus. So now with many other scientific developments like the electron microscopy, as I have already told you, crystallography, it will be possible to share recorded sessions. Yes, we will do it. We'll have to do some editing. Somebody sending a message. So, okay, now now we know that, okay, there are viruses which infect bacteria, and we call them as bacteriophages. But how many, what is the number of bacteriophages we have in this world? So for this, you need to consider some of the estimates given by earlier scientists like Curtis et al., who estimated around 6 million free living microbial species. And then another 3 to 5 million species of single and multicellular eukaryotes appear to have specific prokaryotic associates, was reported by Novotny et al. And now taking both these figures, we can tell they are roughly around 10 million free living and eukaryote associated microbial species in the world. Now, if each of these microbes is a host for at least 10 different phage, then phage species richness is immense. And we can predict that around 100 million phage species. So for this reason, bacteriophages are considered the largest, living entities in this whole world. Now, with the advent of metagenomics, this fact is getting more and more established. Even our gut microbiome, a lot of studies has been done. I think I forgot the figure at this moment. Probably this year itself, there's a paper where it was reported that they have discovered something around 37,000 new bacteriophages by doing some research on the gut microbiome. So we have a lot of marine microbes. You have microbes even in Antarctica, and then apart from that, you have archaea. So you can just guess what is the number of bacteriophages we are having in this world. And now, today we are going to study a lot about the importance of these bacteriophages. And basically now we know that they are parasites of bacteria. They are phages which infect bacteria. So that's why they call as bacteriophages. And you can see their structure over here with a capsid head, nucleic acid DNA, collar, which is tail, basically base plate spikes, tail fiber. This is just one kind, and there are many other kinds. So, and you can see the image under electron microscopy over here. What happened? Okay. So we already know that they are very ubiquitous and abundant, 10 times more in number than the bacterial host. And they're very specific to the host bacterial species, generally limited to one species. The continuous interaction between phage and the bacterial host leads to fight for survival by evolution of their genomes to confer resistance to each other. So these bacteriophages will always try to kill the bacteria. But then there is a contradiction there. If these bacteriophages eradicate away the bacteria altogether, the bacteriophage itself cannot survive in the long run. So there is always some kind of a balance over there, which is still a mystery. And that compulsion probably has, allowed a lot of flexibility on both the parties for the bacteria to evolve and adapt, as well also the virus to evolve and adapt. So it's sometimes a never-ending war, and probably it's also a win-win situation, as debated in the scientific cycles many times. Now, this resistance mechanism is extremely diverse, owing to co-evolving genetic variations, as I was just discussing, and counter-adaptations in both bacteria and the phages. So it is likely that many mechanisms, although we know a lot of mechanisms, some of which we are going to discuss today about the bacterial defense system, how the bacteria adapt to it, but a lot of things are yet to be discovered. Let me tell you a story or take you back to your childhood. So how many of you have read this book, Alice Through the Looking Glass? Okay, there is no any response. Okay, anyway, so here, Bushra Khatun Ansari has told she has read this book. Sonali Kelkare, no. Anyway, no problem. I won't be, of course, spending the time telling the story over here, taking long time. Anyway, so you see here, briefly the story is something like this. The queen was taking Alice along with her, and they were traveling very fast. Okay, now the queen was sounding faster, faster, and so they went very fast. At last, they seemed to skim through the air, hardly touching the ground with their feet. They were actually flying, okay, as you can see. But then, after some time, the queen propped up against a tree and said to Alice, kindly, you may rest a little now. And when Alice looked around, she found out that they have actually not, although they are moving so fast, here in this picture you can see, they have not actually moved any further. They have been in this tree, under this tree for a long time. So she was asking, why do I believe we have been under this tree the whole time, although we were moving? The queen said, of course, it is. And then Alice was puzzled, and the queen told, this is happening because you live in a country which is very slow, a slow sort of a country. So, and the queen suggested, now here you see, it takes all the running you can do to keep in the same place. So the speed with which they were traveling was just good enough to keep themselves under the tree. Okay. If you want to get somewhere else, you must run at least twice as fast as them. So this is the moral of the story in a brief. Although they were running fast, it was not fast enough to take them somewhere else. It was just enough to keep pace and stay below the tree. So now this is something which is known as the red queen effect nowadays. So Lee Van Valen, who is an evolutionary biologist, he used these story to give red queen hypothesis, where he states that to survive, microorganisms must constantly adapt, evolve, and thrive against the ever-evolving antagonistic microorganisms within the same ecological niche. Okay. Now, if we just remember last year when the pandemic first came, towards the end of 2019 and the entire year of 2020, we were all very, very panicked. And because we didn't have any vaccines at that point of time. But then if you see that our scientists worked so hard, many governments around the world had so positive policies. Today we have a lot of various kind of vaccines, and this is a kind of a historical, moment where vaccines has been developed so fast, and they have been, regulatory approval has been given so fast. Because if you would have gone slow, as in the earlier cases, we would have actually reached nowhere. At least we are not there, we know, but at least we are able to keep pace. So this is what is also a principle used by the corporates. And I think it is also equally useful for all of you, for your career paths, or whatever you do. You have to keep running at least to keep pace. And if you want to go to some place, you have to really work double the hard. Anyway, now this concept is required to understand that the bacteria is evolving against the attack of virus. Okay. If it doesn't do, it will perish. It has to keep pace with the tricks the virus plays. Now the virus is also evolving because the bacteria is fighting back, right? So this is a never-ending story in a way, and we never know. There is actually basically no winner. It's all about being in the race all the time. Now, I will not spend much time on the viral life cycle. Many of you are well aware of it, but there are two important cycles, the lysogenic cycle and the lytic cycle. So in the lytic cycle, virulent phases degrade the host genome, leading to the biosynthesis of viral proteins and the nucleic acids for the assembly of phage progeny. The bacterial cell lysed and the newly assembled phages released are ready to infect more cells here. And in the second case, the lysogenic cycle, the repressed phage genome integrates into the bacterial chromosome as a prophage. Proliferation of the prophage occurs during replication and the binary fission of bacterial DNA. Now, amidst this viral life cycle, for both the lysogenic and the lytic, the bacteria has developed certain defense mechanisms. So the bacterial defense mechanism targets different stages of this viral life cycle. So that from that point of view, we need to know what is the lytic life cycle and what is the lysogenic life cycle, because the bacteria has to act against the multiplication of the virus. So it has to stop the multiplication of the virus somehow. Otherwise, that would lead to the destruction of the bacterial molecule. And it also takes a strategy to prevent entry of foreign genetic material and its propagation. Now, these strategies include blocking of phage attachment on bacterial cell surface and injection of phage DNA. Okay, you can see here, number one, prevention of receptor recognition. And you can see that there's a cross mark over there. The virus cannot sit over there because something might have happened to the receptor molecule over here. Anyway, secondly, small molecular chemical production that interferes with the phage or its DNA. Another way is the degradation of the phage DNA by restriction modification, which you already have come to know from Samrat Bora in the morning, or by CRISPR-Cas9 system or CRISPR. Then the activity of mobile genetic elements interfering with viral infection like PLE, phase island inducible islands like elements, and phase inducible chromosomal islands. Now, another method is the dormancy or suicide by infected bacterial cell by adsorptive infection and toxin antitoxin module. Okay, so the virus actually, the bacteria will commit suicide so that it doesn't become a source of infection for others nearby. Then assembly interference and capsids piracy. So these are some of the strategies the virus adopts. So you can see the pictorial representation over here of the various defense strategies. Number 1, 2, 3, 4, 5, 6. We'll come back to them again and again from time to time. So overall, the types of bacterial defense mechanism can be broadly classified into 3 broad groups. Number 1 is a resistance based on variation of virus receptors to prevent phage adsorption. So you change these receptor types, okay? And we also know that COVID, SARS-CoV virus entered our lung cells through certain receptors. So if we are able to mutate those receptors it will not be able to enter. Even in fact, earlier, of course, now the concept is changing. Earlier, it was believed that very young children have very low expression of those receptors in the lung cells, due to which the virus is not able to infect the young children, very easily. But now, of course, the statistics is changing. We actually really know very less things about COVID virus. Anyway, let us focus back on the three types of defense mechanism. The second one is the development of immunity, particularly innate immunity by RM systems, restriction modification systems, and the adaptive immunity by the CRISPR-Cas systems. So within these three broad groups, you can find actually variations. Okay, here you find there are two types of immunity: the adaptive as well as the innate immunity. And I have to tell the non-specialist over here, immunity is something very, very complex in multicellular organisms like humans. But when you tell about bacterial immunity, it is altogether a different concept, not like the immune system of the humans. Okay, it's actually basically a molecular immune system. It's not our immune system is comprised of many things. You have organs, immune organs. You have immune cells. Okay, so many different things are there. It's a very complex system. In comparison to that, the bacterial immune system is very, very simple. It's actually basically a molecular system, but it is very, very efficient, as you will come to know. The third one is the dormancy induction and program cell death, which I already told you. The toxins, antitoxins, and the abortive infections. So these actually help the bacteria fight the FAS attack. Now let's start with bacteriophage absorption. So as we were discussing about DNA binding proteins or nuclear binding proteins, nucleic acid binding proteins yesterday, the proteins and the DNA has to come together and bind to each other to do something, right? The protein has to bind to the RNA, or the RNA will bind to the protein, for example, the Cas9, and then it will drag it to the site of the DNA where it will do its work. So similarly, for bacterial defense to happen or disease to happen, the FAS has to come and sit on the bacteria, or it has to come and absorb. So this is the first step over here. So you can see here in the picture, we are taking two types of bacteria: the gram-positive bacteria and the gram-negative bacteria. I will not go into the chemical details of it, but you can see that they are different. The structures, one is having very smooth structure. The negative one is having some kind of additional features on its surface. The FAS recognizes the receptor binding proteins, and it binds to the host-specific receptors on bacterial cells, which starts the infection process. So that is the first step. So lipopolysaccharides, teichoic acids, and other cell surface structures act as irreversible FAS binding receptors depending on gram-positive or gram-negative bacterial species. These receptors are usually present in the cell wall, bacterial capsules, slime layers, villi, or the flagella. So FAS adsorption is a two-stage process. The first step is the initial reversible binding to surface exposed structures. Subsequently, irreversible attachment to a secondary receptor. Okay, so these are the first two stages. Now here, bacteria goes for preventing the FAS adsorption. So you don't even allow the person to get into your home if he is a troublemaker, right? That is the strategy many people adopt, right? And it's one of the perfect strategy, because once the guy comes inside, you really have to fight with him and throw him out. So you spend a lot of metabolic energy in doing that, and a lot of problems may occur, legal entangles, so many things. So bacteria is quite clever in that way. It will not allow the phage to enter it. So bacteria has evolved to devise a few mechanisms to prevent absorption of the phage on its cell surface. It employs receptor masking. It will mask the receptor or modify it or mutate it, and production of decoys to avoid recognition by phases, right? So it will produce some kind of decoy molecule to which the phase will go and bind, and it will be duped, and it will actually not be able to enter the bacteria. The absorption blocking mechanism basically works based on three different strategies. You already know. Number one is the blocking of phase receptors, production of extracellular matrix and outer membrane vesicles, production of competitive inhibitors. So these are all expressed over here. You can see in this particular figure, bacteria modifies the cell surface receptor to avoid binding of phage RBP to it. Phages can adapt to recognize these new receptors. Bacteria can also produce proteins that mask the phage receptors, like binding of TRAD protein to OMPA receptor in E. coli, which prevents the phage. Staphylococcus aureus produces a protein A, which reduces phage adsorption. Okay, so here you can see in this figure number 4. So you can see some kind of protein A is being produced. Now this actually doesn't allow the phage to bind. Then 50 is the phage adsorption can also be blocked by the production of exopolysaccharides, EPS. Okay, you can see in this figure the EPS being produced. But phages overcome it by producing a polysaccharide lyase enzyme or a polysaccharide hydrolase to cleave the EPS. So you see, as we were telling in the beginning, they fight with one another, and they evolve their own mechanisms to overcome the strategy developed by the other. So here in this case, the bacteria is utilizing exopolysaccharide. The phage is trying to overcome it by producing an enzyme or group of enzymes which can hydrolase or cleave that particular EPS. Now, there is a sixth strategy. Phages have also evolved to specifically recognize polysaccharide portions such as the O antigens and the K antigens. Next is the production of ECM and OMV. Here, the high molecular weight polymers are secreted by bacteria in response to increased phase pressure to form biofilm that makes the surface receptor less accessible to the phage. Okay, many of you may be knowing about quorum sensing, where the bacteria survive in groups as a colony and not as an individual, and they exist by forming a biofilm. Now, some of the many diseases which are resistant to drugs adopt this kind of adaptation technique as well, formation of the biofilm. And the behavior of the bacteria when cultured individually and when it is in a biofilm is altogether different. And that's why any drug which is developed against the individual isolated bacteria fail to work when the bacteria exist in a biofilm mode. Anyway, so here these guys, bacteria are using the biofilm mode to avoid the contact of the phage molecule. So with biofilm, the bacterial cells are specially arranged in a way that outer layer of cells closer to phage are metabolically inactive to prevent phage reproduction while protecting the inner laying cell. So if you look into it, this is a kind of a organization of the colony. So inside in the core, the bacterial cells will keep on dividing, but on the periphery of the sphere or on the surface, the cells which will be there, to which the phage will come and try to absorb or attack, those are metabolically inactive. Now you have to remember the development of the idea or the discovery of the bacteriophage, where they observed that, or the viruses, where they observed that the virus, that infectious agent will multiply only will actively growing tissues. So you need actively growing tissues or actively growing cells for the viruses to multiply. Now, if this virus go and enter an old cell which is not going to divide again, the virus cannot actually divide. And this is being exploited by this biofilm formation. The cells which lie on the exterior are metabolically inactive and they don't divide. Therefore, even if the virus comes in, it won't be able to replicate. Now, what are the components of ECM? You can see some of the things in the figure over here. Now, these components can act as decoy or sink to absorb and immobilize phage that prevents getting near the cell surface receptors. So, example, the Carly proteinaceous components of ECM confers phage resistance to E. coli. Then the polysaccharide capsules encoded by capsule genes are secreted by bacteria that marks the cell surface receptors from phage. For example, there's an overproduction of colanic acid, phage resistance, which confers phage resistance to E. coli. Then outer membrane vesicles, or OMV, or extracellular vesicles containing surface receptors absorbs phage before binding to receptors in Vibrio and Escherichia coli. So another strategy is the alteration of the receptors. The alteration of the primary and secondary cell surface receptors of bacteria is a strategy adopted in the phage resistance in this case. So you know about point mutations, where there is a single base pair change. So here the point mutation in protein receptor genes or in bacterial genes for extracellular superstructures can prevent the phage binding. So in certain cases, a single point mutation actually may change the structure. Okay, so that is good enough to prevent phage binding. The bacteria uses phage variation or reversible switching of gene expression or masking of receptors as a more prominent strategy for the phage resistance. For example, you have the case of bacteroids, which is a gut symbiont that utilizes phage variation in around 19 loci, controlling the production of different capsules and S-layer proteins expression to avoid host-specific phage attacks. Another method is downregulation of the receptors expression through phage variation can be achieved by recombination or predisposed alterations in the promoter regions. Phage variations can induce temporal chemical modifications of receptors, as in the case of Lactococcus lactis. The gene of the cell wall dealination activates its response to phage infection. Now, let us discuss something more about the phage variation over here. You can see here the genomic inversion, and you can see the safflon, which is due to multiple inversions. And here there is a slip strand mispairing, and there is DNA methylation. And you know about the restriction modification system already. So all these leads to the modification of surface receptors, as you can see over here. And the restriction modification system will actually allow the nucleic acid of the phage molecules to enter, but they will leave them inside, as you can see here. Now, the mechanisms that allow high frequency or on-off reversible switching of phenotypic expression in part of the bacterial population are defined as the phage variation. So here, the genomic rearrangements mediated by site-specific recombination, and then slip strand mispairing of the repetitive sequences during DNA replication, then epigenetic modification through DNA methylation takes place, as I have already discussed with the help of this diagram. So this is mediated by specific recombinases. Genomic inversions can silence genes. As you can see here, the light colored ones, by changing the orientation of promoter regions. You can see the arrow direction is being changed over here. Then safflons represent a complex set of interchangeable recombination cassettes that creates diversity at the 3' end of the open reading frame, and it generates many different proteins with alternative C-terminal regions. Slip strand mispairing. During application of repeat sequences within open reading frames create frame shifts, and this methylation of promoter regions can temporarily silence the gene expression. So these are the various strategies being adopted. The fast variation enables bacteria to switch between different types of surface receptors at high frequencies to generate immunity against bacteriophages. Apart from the receptors, fast variation also occurs in restriction or modification systems. Now let us go to the production of competitive inhibitors. So let me take a small break of 1 to 2 minutes. You can see here a peptide with this particular structure over here and sequence over here. So, okay, few compounds that are secreted by bacteria can competitively bind with fast receptors to avoid fast infection. Okay. E. coli FHUA or FUA is an iron transporter and also the port of entry for coliphases such as T1, T5, etc. The antimicrobial molecule microcin, which is a peptide, J25, also uses FUA as a receptor and can outcompete fast T5 for binding to FUA. So when you put this peptide over here, it will bind to this receptor, and when the receptor is occupied, the FAS will be outcompeted in this particular case. The microcin J25 is an antimicrobial peptide which is produced under conditions of nutrient depletion and has a role in microbial competition by inhibiting the growth of phylogenetically altered strains and allowing the bacterium to avoid FAS infections. Let us go to the next strategy, where the idea is to prevent the entry of FAS DNA. Phases after absorption, here the FAS will somehow absorb. I mean, the absorption process, the defense mechanism fails. The phage stops to stop it from absorbing. Okay, no problem. The first line of defense is gone for some reason. So now the enemy has entered the border, and of course, in the case of virus, all of you know that the whole virus particle do not enter inside. Rather, it will inject the nucleic acid inside. So after absorption, it injects its genome by degrading the peptidoglycan layer of cell membrane, and with the help of an inner membrane protein. Now, in the case of prophage, the fast genome integrated into bacterial chromosomes. So if you go back to the lysogenic cycle and the lytic cycle, you will understand the difference between a phage and a prophage. So this confers immunity to bacteria from phage attack by encoding membrane-associated proteins called superinfection exclusion systems. Okay. So in gram-negative bacteria, proteins IM and SP, IMM and SP of this superinfection inclusion or PSI system blocks the entry of the phage DNA. So you can see here in this picture, this SP and IMM is there in the inner membrane. So in their absence, this IMM alone in the second case B, in the first case, there is no any IMM or SP, and you can see the nucleic acid is getting injected. It is going beyond the inner membrane. But whenever in the case B, you see there is a IMM, the nucleic acid cannot enter or pass beyond the inner membrane. It can pass the outer membrane, it can pass the peptidoglycan layer. And when you have the SP conjugated with the IIM, the passage of the nucleic acid is stopped in the peptidoglycan layer itself. So these are the very interesting tricks adopted here. Okay. So IMM through two transmembrane domains prevents the transfer of phage DNA into the bacterial cytoplasm by changing the conformation of the injection site. The membrane protein SP inhibits the activity of the T4 lysosome that degrades peptidoglycan, thus providing resistance to phage DNA entry. Now let us go to the next type of defense system in bacteria, the innate and adaptive immunity systems, which together constitute the bacterial immune system. This system basically deployed a technique of cleaving the invading genomes. It will actually digest the nucleic acid. So when the first type of defense of preventing adsorption fails, and even the second type of defense where you have this SP and IMM molecule alone or SP into together with IMM trying to prevent its entry into the cell. And if that fails, no worries, there are other systems which will take care. Okay, let the enemy come in, we will destroy it somehow. So that's the strategy bacteria deploys in this third case. So, and this falls under the immunity mechanism of the bacteriophage, and it has two arms. What is the innate immunity? The other one is the adaptive immunity. Okay, here you can see in this picture very clearly the phage has succeeded in what it intended to do, inject the nucleic acid inside the bacterial cell. But you can see that's yellow TT circular structure, which is a REase or restriction endonuclease. And then there's a restriction recognition site over there, and it will chew it over there. And however, that same restriction enzyme which digest nucleic acids, somebody was asking in the earlier session how the organism's own DNA is protected. You see here the methylated regions are there. So so there are two components to this system, the restriction system as well as the modification system, which you are all aware of. So that takes care of the self and the non-self, and then the enemy's nucleic acid is very easily destroyed in this case. Okay, once the fast DNA is able to get inside, there are a variety of foreign nucleic acid degrading systems in bacteria that comes into action. Okay, let me re-emphasize. There are a variety of foreign nucleic acid degrading systems. Earlier, we only knew about few, but now a lot of these systems has been discovered over the years, and still we are not very sure whether more novel things lie. Probably they are there because we have so much of diversity of organisms as well as the phages. So these are considered as innate and adaptive immunity of bacteria, which can discriminate between self and foreign DNA by modification and destroy the unmodified foreign DNA by restriction, which you are already aware of. The restriction modification system in many bacteria is one of the prominent innate defense mechanisms. The restriction modification system consists of two enzymatic activities, as I have already told you, a restriction endonuclease and a methyl transferase or MTase. The restriction endonuclease recognizes and cuts non-self and unmethylated double-stranded DNA at specific recognition sites. The MTase adds methyl groups to the same genomic recognition sites on the bacterial DNA to protect self-genome from restriction endonuclease cleavage. So these are the basic things which many of you already know. And you can see that this is the third layer of defense in a way. The absorption failed, the entry was successful, no worries, but the RM system will come into action and it will destroy the nucleic acid that has landed up inside the cell. So that is in brief the task the restriction modification system is carrying out continuously as a part of the bacterial defense system repertory. Now, this is something you probably many of you know, the persons who discovered this system, Salvatore Luria and Mary Human in 1952 and 53. And in the 60s, Werner Arber specifically discovered restriction enzymes for which he was awarded the Nobel Prize. These restriction endonucleases or the restriction enzymes cleaves internal phosphodiester bonds, and these are shown here in the pictures over here. And probably Professor Bora, Samaral Bora was telling you about these site recognitions and also their nomenclature. So your EcoR1 is the first restriction enzyme that was discovered in Escherichia coli, and that's why it's given the number 1. And you can see this typical consensus sequence over here, GAATTC, and you know that these are rotational palindromic sequences, and then you have these specified cleavage sites, and as a result, if any bacteriophage enters the E. coli, this particular restriction enzyme will try to scan for the site where this sequence is available. So yesterday I discussed with you about DNA binding proteins and how the proteins recognize the DNA elements. Probably you remember the various simple things like sliding, hopping, and so on. And you also probably remember about the major groove, minor groove role. And you probably remember about the shape recognition and sequence recognition. So here it is actually the sequence recognition which is in play in identification and destruction of the invading DNA molecule. The restriction endonuclease cleaves the internal phosphodiester bond here, as I have already told you once. The methyl transferase that transfers methyl groups from the S-adenosyl methionine donor molecules to cytosine or the adenine in DNA. So this will restrict the restriction enzyme from cleaving the host DNA itself. So you can see here that in the first case, there is no methylation in the invading viral DNA. So restriction enzyme is acting on it, and in this particular case, it is generating a sticky end. In certain other cases, by other restriction enzymes, you may get blunt ends, you may get 5' overhangs, you may get 3' overhangs, and so on. But here you can see these two A adenosines here are getting methylated, So as a result of these, the eco R1 is not able to cleave this. So this is how the self and non-self is, discriminated. So they are classical restriction modification systems. These systems are actually diverse and found everywhere. In around 90% of the sequenced bacterial and archaeal genomes, you find RM systems, which they have developed over millions of years against the invading phage molecules. There is some kind of scientific debate whether the virus which invades archaeal genomes should be called as bacteriophage. Let us avoid that discussion at this point of time, but let us keep in mind that apart from bacteria, archaeal genomes also have this kind of systems. So there are more than 300,000 known or putative restriction modification enzymes currently listed. So it's a big number. I mean, yesterday I was telling you about those 8 classes of nucleic acid, DNA binding proteins. And if you remember, some endonucleases was one of the dominating ones, right? And you can see here today 300,000 members. I mean, some of them have been really discovered, and some of them are putative, which has been predicted from bioinformatics analysis. So several heterogeneous proteins confer their activity and are classified into the 4 groups. I would not like to go into this classification. I could listen to Professor Bora briefly telling about these 4 different systems. But just to tell you that they are just not of one kind. You can see 4 types, okay? And then generally MT is of type 1 to 3 systems methylate specific motifs in the host DNA, while non-methylated sites in the foreign DNA are cleaved by restriction enzymes. The type 4 restriction endonucleases lacks cognate methyltransferases and cleaves DNA modified by the viral methyltransferases. The mechanism here is a little bit different. The restriction endonuclease is usually more active than methyltransferases and rapidly cleaves the incoming fast DNA, while host genome protected by methylation. However, fast DNA sometimes may be methylated by a bacterial methylase to avoid restriction and enters the lytic cycle. So this is a deviation from the normal design. So biology is, that's why very, very interesting. I mean, the good laws, they tell, are always made with exceptions. I mean, you apply that law to a large number of the population, but in certain cases, you need to bypass that law for various regions, which may be humanitarian regions or some other technicalities. So you always create an exception in framing laws and rules. Okay. Anyway, now here you can see how this methylation is being adapted to avoid restriction of the fast DNA also, and pass it to the lytic cycle. Now, this type 1 RM system contains 2 MTAses subunit, 2 restriction endonucleases, and 1 recognition subunit. And you can see here in this figure the 2 HSDR and the 2 MTAses over here. So this modifies both strands of bipartite asymmetric DNA site. 2 restriction complexes bound to non-methylated sites through ATP-dependent DNA looping cleaves at the site in between. It recognizes the methylates and methylates a single sequence, but cleaves DNA up to 1,000 base pairs away. So type 1, it will recognize in one particular consensus site, but its binding site and its cleaving site are lying around 1,000 base pairs away. So here we know that it will bind to this particular sequence, but we don't know where it will cut. Okay, so that is the uniqueness of this type 1 restriction modification system. As a genetic engineering tool, this is not useful to us because for genetic engineering, we need specific cuts, a DNA which is cut specifically. But from the bacterial defense point of view, this is fine because the whole idea is to destroy the invading DNA. So there is no any problem regarding the type 1 RM system. Rather, it's quite advantageous because if your enzyme is very specific in its cleavage, you have large problems. It will only cleave if that site is available. But in this case, you have that advantage. I mean, the cleavage is not related with the binding in a way. In type 2 restriction modification system, you have separate MTAs and RNAs units. Both recognize the same specific 4 to 8 base pair long palindromic DNA site. They cleaves within or near to the non-methylated site. In a type 3 RM systems, here this is formed by multi-protein complexes consisting of modification and restriction subunits. This modifies only one strand of the asymmetric recognition site, and it cleaves at a fixed position from one of the recognition sites when one activated RC interacts with another bound to the non-methylated site. So this is the difference between the type 3 and the earlier two. And when it comes to the type 4, it lacks a modification module, as I already told you, and cleaves DNA after recognition of the modified sites. Now, what are the anti-restriction strategies of phage? The phage may lack endonuclease recognition sites in their genomes through the accumulation of point mutations to escape the restriction, because the virus also keeps on evolving. So it may become very clever. It will just insert a point mutation or pick up a point mutation. And because of the specificity of binding by restriction endonucleases or cleavage, the virus particle will be able to escape. Some phages evolve to acquire the cognate methylase gene in their genomes to become restriction resistant. So this is very clever of them. So they will hijack the MTase gene over there, and then it will methylate its own DNA before pushing it into the bacteria. Phages like polyphase P1, while injecting a DNA along with the host genome binding proteins, DAR A and DAR B, marks the RM recognition sites. In figure C, you can see that the host genome binding proteins. Now, when you look into figure 4, you can see that OCR, which is a protein expressed by polyphases T7, mimics the DNA phosphate backbone and interfere with the RM system. Okay, so in E, you can see the RAL protein of polyphase lambda can stimulate bacterial modification enzyme activity to recognize phage DNA as self. Peptide STP of polyphase T4 disrupts the structural conformation of the RM system. So these are various, strategies which is adopted by the phage to avoid restriction. Okay, and then thereby this will lead to the failure of the bacterial defense system. So here you can see there is an infection in the classical restriction modification system, and in B there is no infection. You can see binding of some molecules over here, and then in C you can see again there is some infection. because these molecules which were restricting it are not there or defunct. In the case D, you can again see the infection. And in case E, you can see there is again no infection. So let us see in detail. And then you also see in figure C, and there is a molecular SMC, and in figure E, there is a gluco-MS SMC. Okay. So the genome of FAST-T4 contains the unusual base hydroxymethyl cysteine instead of the cytosine, which makes it resistant to restriction modification system that needs cytosine in specific recognition sequences. Few bacteria evolved to contain modification-dependent systems such as the DPN1 in Streptococcus pneumoniae and MCRA, MCRBC, and MRR from E. coli to indiscriminately recognize methylated or hydroxymethylated DNA. This FAST4 has also in turns evolved to glucosylate SMC, hydroxymethyl cytosine, to confer resistance to MDS and can infect a bacteria. So you can see in the last case, the infection is happening. Now, in few E. coli strains, they have prophage that encodes two component systems consisting of glucose modified restrictions and glucose and GMRD proteins that specifically recognize and cleaves DNA containing glucosylated SMC. Hello. Some voice interruption is there. There's some background voice coming. Rahul Karmakar is speaking. Okay, Rahul, your mic somehow got switched on. Okay. Anyway, no problem. Yeah. So now there's an internal protein I. You can see in figure G, IPI encoded by few T4-like phages, which is co-infected along with the first DNA to interact with the GMRS and GMRD complex to inactivate its restriction activity and infects the bacteria. In other E. coli strains, they produce a translational fusion of GMSR and GMRD that can render these IPI ineffective. And still few FAST-T4 mutants can bypass the GMRS and GMRD fusion by unknown mechanism and infect bacteria. So that's a little bit of exception over there. Now you have various other restriction modification systems like phase growth limitation system. This was first identified in the Streptomyces colicolor in RM-like defense systems in bacteria against FAS. So I'm not going to discuss this system in depth because we have few more interesting things to. Rahul, who is Rahul? Let me check what is happening. Rahul, how the microphone got. Rahul Kumar. Rahul Kumar, are you actually attending? Rahul Kumar is switched off. Just a minute, let us take a small break. I will try to check out. Okay, there was some disturbance. I don't know, somehow somebody's microphone got switched on. I think it is now solved. Okay. Thank you, all of you. So there are other systems like the bacteriophage exclusion systems of Brex, which was discovered in 2015. And you can see that these are, not that very old, just 6 years ago. These were discovered. So we'll not go into the details of Brex system at this moment. But you can just see that the core components of this type 1 Brex system include BRXX, which is an adenine-specific methyl-transferase. Then BRXZ is an alkaline phosphatase, and BRXC, which is an ATPase. Then BRXL, a lawn-like protease, and the BRXB is a small protein of unknown function. So this whole system works in collaboration with one another of these components. So in the type 4 Brex, methyl transferase is replaced by a PAPS reductase, an enzyme that can be involved in DNA phosphorylation. Now, there is a concept of defense island system, which we will discuss later on as well. So this defense island system associated with restriction, modification, or disarm systems was identified in Bacillus in 2018. This is very, very new. So disarm type 1 comprises 5 components. One is the helicase domain, the DRM A you can see in the picture, okay, which is in the pink color here marked as H. Then second component is SNF2 like ATPase helicase, or DRMD, which is A/H in the first box. Then you have the protein with unknown function domain, DRMB. That function is still to be ascertained or assigned. Then the fourth component is the phospholipase D, PLP. This is a DRMC, okay. Then adenine-specific methyltransferase, DRM MI. So this is the composition of the type 1 disarm system. So you have a type 2 disarm system. If you just compare the two figures, you can see there are certain things which are common. DRMA, but the DRM E is there, whose function is not known, as well as DRMB, whose function is not known as well. And you have a variation over here, the DRM M2, represented by CM. So here, DRM A is replaced by unknown function domain DRME, and DRM M1 is replaced by cytosine-specific methyltransferase DRM M2. So this is the difference between these two. So these are recently discovered systems just in 2018, and you can see they also are falling into two types. Desamethylase modifies host CCWGG motifs as a marker of self-DNA like RM systems and restricts incoming fast DNA. The DISM offers protection against phases that lacks the sequence recognized by the RM-like system. Now, the adaptive immunity. So we were discussing about the innate immunity, where the restriction modification takes place. And you can see that restriction modification or the innate immunity is not just comprised of one system. There are many. And as recent as 2018, you have seen that disarms has been discovered, and they fall into two types. And actually many labs around the world still working, and they may find out many different kinds of such innate immunity in place. Let us move to the next most important defense system in bacteria, the adaptive immunity. So CRISPR-Cas9 actually will be delivered at length by another speaker. And I already mentioned it to you in yesterday's DNA binding protein class, nucleic acid binding protein class, and where you have, proteins which binds to RNA. And you have proteins which binds to DNA. And when it comes to CRISPR Cas9 system, you are, having this importance of the RNA binding to protein. And you can see here the guide RNA over here. Just a little bit of, discussion regarding the RM system, which act as innate immunity. Here, the CRISPR-Cas is actually an acronym. It stands for the clustered regularly interspaced palindromic repeats and CRISPR-associated proteins, and this is an adaptive immunity component. Somebody is sending some message. Here's a question. We will take up the questions at a later stage. Okay, thank you. Like higher animals, bacteria can preserve information about previous encounter with phage and utilize for defense in later infections. So this will be discussed at length when the CRISPR system is discussed. Some statistics that CRISPR is found in 40% of bacteria and 90% of archaea. Okay, so it's a very widely used defense system. Upon infection by virus, the viral DNA is acquired and integrated into a CRISPR sequence. So that loci, the future infections leads to targeted cleavage by the double-strand breakage of the invading viral DNA, having a similarity to integrated spacer sequence in the CRISPR. So basically this is the outlay of the CRISPR-Cas9 locus in bacteria. So you have here the CRISPR associated genes, and you have here the CRISPR array. You can see there is a leader, and there are certain repeats over here, and these repeats are separated by certain spacers. So this CRISPR-Cas9 locus contains three parts: array of repeats and spacers, operon of Cas genes, encoding Cas protein components, and the leader portion, which is AT-rich region. The CRISPR array are tandem repeats and spacers. The repeats comprise the same sequence, and the spacers comprise different sequences. And these different sequences are derived from viral DNA. So each spacer means how many different kinds of virus has attacked these particular species in the past. That is some kind of information which is kind of recorded in this CRISPR-Cas9 locus. So it's a kind of a registry, molecular registry, and this record is actually picked up from the sequence of the invading viral DNA itself. Okay, so a part of that will be copied, and there are mechanisms by which they do it, which part they will copy and retain here as a record. It's a kind of a some citizens coming into another country where the visa officer will keep the documentation. Okay, so these repeats are basically originating from the virus. Okay, and they are 28 to 37 base pairs long, though there can be as few as 23 base pairs and as many as 55 base pairs. These are the variations which often happens in biology. So I will not discuss about this discovery in length because you have a full class on CRISPR-Cas9 system, and probably Dr. Rubul Maut will be delivering that lecture from USA. Okay. Now let us go to another bacterial defense system where the prokaryotic organoids PAGO mediates some kind of interference. So here, if you look into the figure, you can see in step 1, invader DNA enters the cell. okay, and it propagates. The propagation of the invading DNA will take place. Then there is some DNA chopping happening over here in stage 2, and there is some kind of a short double-strand DNA fragments taking place, and this double-strand DNA gets loaded over here, and then in the fourth stage, this DNA-guided interference with single-stranded invading DNA will take place, and you have a partially unwounded invader DNA over here. And then you finally have DNA-guided interference with double-stranded invader DNA. So there is another mechanism, which is the dormancy induction and programmed cell death. The bacteria possess systems that can cause cessation of the host metabolism. We call it as bacteriostatic effect, or programmed cell death, bactericidal effect in response to the FAS infection. So there is abortive infection, or ABI, and toxin antitoxin systems, TA systems, which are the two altruistic action that prevents fast replication and stops spread of the infection at the community level. So some of the bacterial cells will commit suicide so that their community is safe. Okay, so they don't allow the kind of replication or propagation or the spread of the virus. So even in the case of COVID, if you remember, the protocol is to go for isolation so that you do not pass on the virus to others. But in the case of bacteria, what they do, they will actually destroy themselves and then save the community. Okay, okay. Now that's kind of an altruistic mechanism that the bacteria adopts. These abortive infection systems are mechanistically very, very diverse. Generally, the ABI systems are composed of two modules. The first is the sensing module. The sensing module will sense the FAS infection and transfers the signal. Second is the effector module. It shuts down host metabolism and or causes cell suicide after receiving the signal. Effectors can disrupt essential cellular processes such as translation, transcription, and replication, or by inducing membrane leakage. The mechanism of infection recognition to trigger ABI is not fully understood yet. So here you see the general principle of abortive infection responses. The effector PRRC RNase also inhibits the translation by cleaving the tRNA after activating by peptide STP of phase T4. The RNLA RNAs carry out the cellular mRNA decay. In the second stage, you can see over here, as FAS infection interferes with the continuous gene expression. Then comes the STK2. This is activated by the FAS, cyanM1 protein pack K, and phosphorylates phosphoproteins inhibiting the core metabolism. The REX-B forms an ion channel leading to the loss of membrane potential after sensing viral replication complexes. So this induces cell dormancy or suicide. The phase burst is kind of hampered. Among the many ABI systems, cyclic oligonucleotide-based antiphase signaling system, or CBUS, abortive infection system is recently characterized. So this figure taken from Lopatina et al. 2020. So this CBUS ABI includes an oligonucleotide cyclase, and an effector gene. The oligonucleotide cyclase, called as CGUS, you can see this yellow picture, yellow structure over here. This is activated during phase infection, and it becomes active CGUS by an unknown phase component. So the phase releasing one component after the phase infection takes place. On the extreme left side, you can see it is releasing some component. That component comes over and binds to the CGUS and activates it. And this produces cyclic oligonucleotides. So production of the cyclic oligonucleotide activates a cell-killing effector protein. So once the oligonucleotides will be cyclized, it will activate a cell-killing effector protein, which depending on the CBUS type. What is this particular CBUS? And can be a phospholipase, a nuclease, or a membrane spanning protein. So you see this phospholipase is getting produced here by this cyclic oligonucleotide active effectors, or the DNase is getting produced here, or a membrane spanning ion channel will be produced here. So these will all lead to the abortive infection. The next is the toxin antitoxin system. The toxin antitoxin antitox systems are comprising a stable toxin subunit and unstable antitoxin. Antitoxin is labile and must be continuously expressed to neutralize the toxin. In this pair, the toxin in mRNA form is neutralized by a non-coding mRNA or protein antitoxin, and a protein toxin is neutralized by the small mRNA. The toxins possess various catalytic activities, including DNase and RNase, or can inhibit the DNA replication, ATP synthesis, or cell division. Under stressed conditions, increased antitoxin degradation releases activity of the toxin, leading to the growth arrest. The TA modules are frequently found within the defense islands, and there is an extensive domain exchange between the TA and ABI systems. TA has a high genetic diversity containing at least 6 TA types, categorized based on the nature of the toxin and the antitoxin, protein or RNA, and the mechanism of the toxin neutralization. In addition to the fast defense, they are involved in stress response, plasmid maintenance, and persistence cell formation. So let us have a look into the general principle of the TA system. So you have a toxin in the red color over here, toxin gene, which produces a stable toxin, and there's an antitoxin, which is unstable. The phase interferes with antitoxin expression. Okay, so thereby from this TA system, the toxin is released. So release of the toxin activity happens. So that may lead to the membrane degradation, or you can see some kind of nucleic acid degradation, or stringent response where you have translation inhibition. So here, Hoc toxin causes cell death by depolarization of the membrane, as antitoxin shock and antisense RNA degraded by the phase infection. Toxin and RNAs after degradation of antitoxin due to phase infection degrades host and phase transcripts, so there will be no any translation further. Toxin SAS, small alarmon synthesis, can cause growth cessation, including starvation response, starvation stress response. So we know that for the bacteriophage to survive, it has to leave the bacterial cell after the assembly of the bacteriophage. And then it will kill the bacteriophage and it will be burst, the bacteria will be burst, and bacteria will be killed, and the bacteriophage particles will be released. So now, one of the strategies is to interfere the bacteriophage assembly. You do not allow the bacteriophage life cycle to complete. The replication of the nucleic acid may be there, the synthesis of the protein capsid structures may be there, but you do not allow them to come together to somehow stop the assembly. So phage inducible chromosomal islands are mobile genetic elements randomly transferred by phages to bacterial genomes through transduction. They can hijack phage DNA packaging and machinery and capsids to load their own DNA, a process called molecular piracy. These PICI genomes are often small, encoding genes required for accession and integration factors that promote PICI packaging and dissemination. A repressor generally inhibits their expression in gram-negative, gram-positive bacteria. How an anti-repressor is produced by helper phage during early during the lytic cycle after their infection, and PICI expression is induced. Suppies, the S. aureus pathogenicity islands, then PICI in Staphylococcus aureus has been characterized. Suppies are induced when their repressor STL is sequestrated away by an anti-repressor. The PICI genome replicates and expresses proteins that repress later helper phage genes and alter the phage capsid size to be more appropriate for the PICI genome. This in turn leads to both the preferential packaging of PICI genomes and the prevention of the formation of helper phage variants. In contrast to gram-positive bacteria, gram-negative PICIs are induced by a PICI encoded activator whose expression requires the helper phage. So phage-inducible chromosomal island-like elements. The phage-inducible chromosomal island-like elements are mobile genetic elements found in Vibrio cholera. These PLE are similar to other gram-negative PICIs, however, gene content is little different. Upon infection of the host bacterium by the phage, the PLE encoded recombinase INT accesses PLE from the Vibrio cholera chromosome. PLE then replicates to high levels and inhibits ICP1 phage replication by unknown mechanisms. However, several ICP1 phage isolates encoded their own CRISPR system capable of neutralizing PLEs, allowing ICP1 propagation in PLE positive Vibrio cholera strains. So with this, we have covered almost all the various known bacterial defense system against bacteriophage till now. So you can see in this figure as a revision what are the various systems you learned in a way. So we learned about the innate immunity system where you have restriction modification, BRAX, DND, and disarm, etc. Disarm has been, recently known. Then you have this adaptive immunity or the CRISPR-Cas9 system. And then you have certain chemical defense where you have inhibition of the phage replication, DNA intercalence, okay, and then enzymatic pathway anyway. Then there are certain abortive infections that you have seen, abortive infections where the kind of suicidal phenomena takes place due to the altruism of the bacterial population to save the community. Then you have the signaling system, the CBUS, and of course there are many other unknown mechanisms. Apart from preventing absorption of the virus of phage molecules onto the bacterial surface. So we were discussing about some defense islands in between, not discussing, rather mentioning about the defense islands. So you now know there is always a competition between the virus and the bacteria to survive, and they deploy various mechanisms. For example, the virus picked up the methyltransferase gene through which it methylates itself. Okay, so there is a constant arms race leading to the evolution of antiviral defense mechanisms, as well as strategies adopted by the virus to overcome this defense system. So recently, I mean, it has been found that the immune arsenal of bacteria against bacteriophage is much more diverse than the previously envisioned. Although we discussed these few defense systems, actually we don't know much about this antiviral defense system in bacteria, and a lot of things remain to be discovered. Up to date, whatever discovery has been made, there are certain contradictory observations. For example, the individual microorganisms often encode multiple distinct defense systems, some of which are acquired by horizontal gene transfer, alluding to their fitness benefit. And in contrast to these, defense systems are frequently lost from prokaryotic genomes on short evolutionary timescales, suggesting that they impose a fitness cost. Okay, so some of them they acquire it, some of them they lose it. So there is a gain of some kind of genes and loss of some kind of disease. You have a fitness benefit in the first case and a fitness cost in the next case. Why will they incur this fitness cost? Okay, so this contradiction is kind of addressed with the concept of pan-immune system model. This is something very, very interesting. I will just briefly discuss before we come to a conclusion of today's lectures. So an effective immune system is not the one encoded by the genome of a single microorganism, but rather by its pan-genome, comprising the sum of all immune systems available for a microorganism to horizontally acquire and use. So this means that in a particular ecological niche, certain bacteria and their related species or types, they will try to possess all these arms or arsenal as a community, on a community ownership basis. So every house may not have all the gadgets, all the equipments required. But when you require a particular tool, you go to your neighbor's house and bring that tool. Or sometimes you don't have a screwdriver to fix your, electric bulb because you forget to, or you didn't felt you should buy it, or somebody may not have a hammer in his house. No problem, but you know that another person in your neighborhood has that hammer. You go and take that hammer from him, do your work, and maybe return it back. And sometimes it may happen your friend may not want it back, and he may ask you to retain it, whatever. So immune system is also something like that, because bacteria is a very small organism, and then carrying all the defense systems along with it is a kind of a genetic load, and also metabolically it's very, very taxing. So that's the reason why at certain instances, certain bacteria will go for this loss, and whenever it is required, they will try to acquire it, and the horizontal gene transfer mechanism facilitates this very, very easily. So you will find that the antiviral defense system tend to cluster on bacterial and archaeal chromosomes, and these regions are denoted as defense islands. So it's something like the army living in a cantonment. you have all the different kinds of army, whether the infantry or the paratroopers, all living in the same cantonment. So and maybe sometimes the Air Force, the Navy, wherever for strategic regions they may live in the same locality for logistical strategical purposes. So in bacteria also, all these defense systems, they tend to remain in the same genomic loci. Okay. So these defense islands typically comprise diverse defense systems. So you can see in this case, you have this recombinase CRISPR-Cas3 system, the TA toxin antitoxin system, Doria, and then RM1 system in this particular sulfovibrio case. Then the bacillus, you have this transposable, then Gabija, then restriction and then nuclease, and so on. So you can see from this, and then you have in certain cases the abortive infection related genes over there. So these defense systems are enriched with genes typical of mobile genetic elements such as transposes, recombinases, and conjugation genes. So this helps them actually in the mobility from one bacteria to another bacteria through horizontal gene transfer. So some of these defense islands were predicted to encode more than hundreds of different genes. Okay, so this mechanism of formation of defense islands are currently unknown, but there are certain assumptions that co-localization of defense genes with mobile genes could facilitate a horizontal transfer of multiple defense systems from one bacterium to another in a single transfer event. Okay, so these islands can be hotspots for integration of horizontally acquired genes with the defense systems clustering in defense islands through the garbage and pile effect, in which high rates of acquisition and loss are not strongly deleterious. In addition to these, such co-localization of defense genes could reflect functional links between the defense systems, including possible co-regulation or positive epistasis. Okay, so this epistasis also is something very, very interesting concept cupping in this bacterial defense system context. So a bacteria may have, for example, in one case you are finding out it has 100 defense systems or 100 defense genes. Okay, and these 100 defense genes may belong to say 10 or 20 different defense systems. Now, does the kind of activity of one defense system mask or inhibit the activity of another defense system? This is becoming an interesting area of research. So this was something, the genetic epistasis, if you remember. So this is kind of a epistasis at the systematic systems level. So if one system, suppose the Air Force goes out and the Army don't need to really go, the Air Force will go first, and then it will do some kind of attack, and then the Army is sent at the later stage. So those are part of the defense strategy. And the bacteria has to optimize the metabolic energy, right? And for that reason, it may not want all the defense systems getting activated at the same point of time. So these may operate at the systems level epistasis. Now, so you know that there are defense islands in the chromosome of the bacteria. So where in these islands you find many different defense systems, and these defense systems have many multiple genes, which may be as high as 100 number of genes. And you now also know that not all the bacteria in an ecological niche will contain all the defense systems. So they will try to acquire the defense systems they do not have whenever it is necessary. And in certain times, they will try to give off those defense systems. There is a kind of a loss and kind of a gain from time to time. Now, also in my discussion, I have highlighted many times, we know very little about the bacterial defense system, although so many things has been discovered over the years. And it actually, I have actually avoided discussing the known systems in detail because of shortage of time. But you have to remember one last thing, that a lot remains to be discovered when it comes to the bacterial defense system. It's an active area of research. Many of you may really think of undertaking it. So now, with this concept of the defense island, you have a big opportunity of discovering new defense systems, right? So probably you know about this famous quote by Aesop's, a man is known by the company he keeps, right? So taking these as an inspiration, if you start exploring the defense islands, okay, you are most likely going to discover new defense system. You really don't have to look something in the dark. You just need to focus on the defense islands of the bacterial chromosome. And there you have certain things which is known and a large number of things which is still unknown. So here lies the scope of this future discovery of defense islands. This is just to show you some data that closely related bacterial strains encode diverse defense system. You already know about it, but just some data from the Natural Reviews Microbiology. And you can see that E. coli, for example, so many different kind of strains are there. And you have on the x-axis the various defense system, RM1, 2, 3, 4, Brex, Disarm, CRISPR-Cas, 1 to E, and CRISPR-Cas 1 to F, Gabiza, and so many CBAs. But not all of them have all of these systems. Some of them have many different kind of systems. The empty boxes means they don't have these. The colored boxes mean they have these systems. Okay, and some of the systems are very dominating. For example, in E. coli, you find it RM2 is present almost in all these known strains. But of course, you may find out a strain where the RM2 may not be available. As you can see in the Pseudomonas aeruginosa W3662, it do not have the RM2. Okay, so this is just to tell you about the diversity of the defense system. And then every bacteria may have only some of these, and some of them may have a large number of these. And what is important is the community ownership, what we call as the pan-genome defense system. So this is the maintenance of the diversity on the one hand, and the gain and loss in the pan-immune system, which we all discussed. So various papers were, considered for preparing this talk. So the references are over there. And I have to acknowledge Mr. John Juti Kalita, my PhD student, who helped me a lot in preparing this lecture by collecting the various literature and also the figures. So I also thank AICTE once again for funding this, sponsoring this particular workshop. Now we can have some discussion. I will try to take up the questions. Some questions were popping up in between. So yes, let me check. Let me scroll through. Yes, sir. So Ajay Kumar Das is asking, bacteria evolves themselves for their defense. What is about the evolution of foreign DNA that used to invade bacteria? Okay, I think where I have addressed this question in some way, but of course now as you are telling, what about foreign DNA that invade bacteria? Of course, DNA themselves cannot invade. It is actually the viral DNA, right, or viral nucleic acid, which invades the bacteria. So I already told you one example. Some of the virus, they have stolen the MTAses and methyl transferases, and then they have methylated their genome. So when they pass it on to the target host, the restriction modification system do not work over there, and the virus survives. And it goes to the lysogenic cycle by bypassing this RM system. Okay. Sir, does production of hydrogen sulfide by bacteria comes under defense system? I am not very sure about this. But probably hydrogen sulfide is a byproduct of a metabolism. Okay. You may have to help me by telling whether the presence of hydrogen sulfide kills the bacteria. If the hydrogen sulfide will kill the bacteria, then it will come under the method we described, the abortive, infection or suicide, right? So if the not only hydrogen sulfide, any metabolite which the bacteria produces to destroy itself, it will come under the defense system. So what could be the probable timeline of this wonderful symphony of competitive evolution? Yeah, it's a question of evolutionary biology. I think it's over millions of years, and it's quite interesting evolutionary biology. Basically, it's a co-evolutionary biology where both are trying to harm one another, but in the process they are also strengthening one another in a way because of the co-evolutionary process. And as humans, what we are trying to do is trying to exploit this kind of phenomena for our own advantage. So, for example, the whole purpose of putting this lecture in here is to give you some kind of a basic idea about the bacterial defense system and how diverse it is, number one, so that when, and it is an application side, for example, the restriction modification system, we use it largely in the genetic engineering, okay, number one. And number two, when we are discussing about which is part of the innate immune system. And now when we are discussing about the adaptive immune system, the CRISPR-Cas9, so you see that we are today using for genome editing, right? So now these are the two known applications. And then the third part is the horizontal gene transfer idea. Now we use this horizontal gene transfer in transferring foreign genes, right? And you have these bacteriophages or the virus injecting the DNA into the host organism. So from that, we have taken the idea that we can use the viral DNA for transferring genes into host organisms. So overall, this lecture is a foundation for understanding those techniques at a later stage. Yeah, so what is the next question? Yeah, Richard Dawkins. I mean, these are, you see, ideas provided. Obviously, I mean, nature probably do not evolve in a very planned way. Okay. I mean, it's a matter of big discussion. These are philosophical approaches where people put their own viewpoints, and very, very interesting viewpoints. So these another kind of a popular thought that the nature do not select the best. Okay, if nature would have selected the best today, not the Homo sapiens, it would have been the Neanderthals who have been dominating here, because they had the better features than the Homo sapiens. They were much more stronger, right? So nature probably go for some kind of statistical averaging. Okay, right. Yeah, thank you, sir, for your excellent deliberation. Yeah, how does RM system discriminate methylate its own chromosomal DNA at the same time? Who is asking? Rajasekhar saying, yeah, I think it's well explained, the methylation and the self non-self concept. At the same time, it doesn't work on. Uh, at the same time, it doesn't work on viral invading DNA. Now, it will work on viral invading DNA, but the viral virus or phages which have picked up the methyltransferase, stolen the methyltransferase genes and made part of it, and where it has methylated, its own DNA, and the DNA methylation actually has to happen in the host in the prior circle, because inside the virus, the methylation cannot happen because there is no any cytoplasm or a biochemical reaction space over there. So the virus acquired it, made a part of its expression cassette, that whenever the viral gene replicates, it also has its set of methyltransferases acquired, or it may use this host system methyltransferases also. So it methylates its own DNA as a part of this defense mechanism, get it packaged, and then it comes out of the bacterial system. Now, these particles, when they re-invade another host, the host will not be able to recognize the viral DNA because it has been methylated. So what could be the probable timeline? So the blind watchmaker, the chromosomal DNA doesn't work. So restriction part of RM system works, recognizing restriction site. Somebody is writing, nature favors the luckiest. So it is coming from my other account, which Samrat Bora is using. Probably you typed in something? Okay. He could not log in due to some reason. Okay, right. So let us stop for today because we have another lecture in the second half. So let me check out the schedule of the next lecture. I think it is at 2:30 or 2:30, right? Yes, it is here. Okay. No. Yeah. Oh, yeah. So next lecture is at 2:30 p.m. Okay. And Professor Ranjan Tamuli will deliver his lecture on talents. So we'll meet at that point of time. Okay. So thank you, all of you. And second half probably, okay. Let me check with Professor Tamuli. Thank you, all of you. What is the attendance? Attendance to Kimano, sir.